Hi everyone, welcome to the June edition of SciBar. June, so we're almost halfway through the year, and obviously most of you will know that some of the lockdown restrictions are still in place, so we're coming to you yet again virtually. We heard this week, in England at least, that from the 4th of July some of the pubs and cafes will be opening, which is great. It means we're one step closer to being able to present to you in the flesh. But I will point out for now that certainly for our events of Palace of Science in the immediate future, we will still be presenting online. For anyone that's seen our events in person, you'll know we like to cram a lot of you into a small event space so we can all have a drink and a discussion together. And unfortunately, as COVID still is out there and a risk, we're going to hold off doing it in person until we can be sure that it is safe. But on the bright side, it does mean that you can enjoy our events in your own home or in the garden in this lovely weather. And you can also enjoy the beverage of your choice, which I hope you are doing. And it also means that all of our events are being recorded. So if you want to see any of the stuff we put out over the last few months, or if you want to watch this event back again, if you enjoyed it that much, then you're able to go over to the Palace of Science YouTube channel and catch up on everything there. So for anyone that has been to our events in person, you will know that the social side of things is really important to us. And obviously it's quite difficult to capture online, but we do try. So you will see there is a chat box on the right-hand side that you're able to write your comments in, like this, hopefully, is me. And at the risk of sounding a bit like Cilla Black, I will encourage you to tell us who you are and where you come from. You will need a Twitch account if you would like to post in the chat box, but it's free and it's easy to sign up. And it does mean that you can type any questions you have in there for the speaker tonight as well. So, as many of you know, SciBar is our monthly event where we get a scientist or researcher from the Northeast to come to talk to us about their work. So normally this is a really informal event, usually at a venue that has a well-stocked bar, but we're making do as is. And SciBar is brought to you by Palace of Science. We are a not-for-profit organisation operating in the Northeast. And to summarise what we do, essentially we sit around thinking about all the awesome science that's going on in the Northeast and ways that we can bring this to you, the public, to enjoy. So I should point out at this point, everyone that works for Palace of Science works. We're all volunteers. So when we're sitting around thinking of these events, usually we should probably be doing something more important. So for those of you that don't know, I'm Nicola. I am one of the events organisers. And SciBar is mostly brought to you by my procrastination. So, good things do happen. So tonight you're going to hear about some of that awesome research happening in the Northeast. But before we get to that, I'm just going to give you a very brief overview of how this is going to run for anyone that hasn't joined before. So in a few minutes, I'm going to hand over to our speaker, Dr Paula Salgado. And she's going to talk to us for about 30 minutes about some of the work that she does. Tonight's talk is going to be extra interactive, so just make sure that you have your phones to hand. There is a website that you should have seen advertised, but Paula's going to mention it in her talk as well. It's menti.com, and you'll be asked to put in a code to join some of the questions that she's going to be asking. So just for anyone that has tried logging in earlier, the code might have changed. The code is now 4469. But again, Paula will cover all of this in her slides. After the talk, there is going to be a 5-minute break, and we actually have something new for you in this break as well. So I've put together a few pub quiz questions for you all to try. So it's 10 questions on science and general knowledge. I think I've made them relatively okay, so I'm expecting most people to get about half right, but of course you can tell me what you get in the chat. And after the quiz, we will come back for a question and answer session. So all throughout the talk and throughout the break, the quiz, you can type in your questions in the chat box if you have any for Paula. So tonight's presenter, as I have mentioned, is Dr. Paula Salgado. She is a macromolecular crystallographer at Newcastle University. And Paula actually presented some of her work at one of our events earlier this year in January. It was a Women in STEM event at the Lytton Phil in Newcastle. Back in January seems so long ago now. And we were just discussing actually how funny it was at that event. Just before she gave her talk, she mentioned that she was hoping no one would ask any questions about the coronavirus, because if you remember back in January, it was sort of kicking off in China, but we didn't have much over here. No one really knew that much about it. Obviously now we know a lot more about it. I think we all do, Paula included. But I am mildly curious if there's any sort of topics Paula is hoping to avoid tonight, if there's any heads up she can give us on the next global crisis like she did back in January. I'm sure that's not the case. But that is everything from me, and I will now hand over, if you can all give a warm virtual welcome to Dr. Paula Salgado.
Thank you very much, Nicola, and thank you to Palace of Science for the invitation to do this. This was, as Nicola said, this was meant to be in a bar. There's a lot of things that I had planned when I accepted the invitation that are just not possible. I will still try and make this as interactive as I can. If it's for you, if you just so you know, I'm actually staring just at my computer. I normally like to have a nice interaction with my audience, so this might be trickier, but I'll try. I'll do my best. And as Nicola also mentioned, one of the ways that I'm going to try and get you to join in the conversation is there's going to be a few questions to answer throughout the talk. So let's start with that. So if you want to join in, you should just go to this website, menti.com, and type in the code 4469. And this is the first question. So have you heard of antimicrobial resistance? And when I'm saying antimicrobial, it can be antibiotic resistance as well. And I'll explain what those things are later on. So I'll give you a few minutes to log on to the website and also to answer the question. And in the meantime, I can start telling you a little bit about what I'm going to do today, which is I'm going to give you an idea of, I'm going to focus on antimicrobial resistance, and then I'm going to specifically talk to you about the work that we do in the lab on one of the bacteria that is involved in this. So that I hope that most of you, I can see about 6 people have answered, so I'm going to give it a little bit longer for people to log in and see the answers coming in. I'll wait until there's at least 10 of you that have given us an answer. Have you heard about antimicrobial resistance before today, or before you heard about the topic of today's talk? And in a second, we'll get the answers. And we're starting to see there's basically everyone has. That's really good. Every one of the 9 that answered the question, 10. So that's really good, and that gives me some confidence about what I'm going to talk to you about today. So we'll move to the next question, which is, so you've heard the term antimicrobial resistant, but who's resistant to antimicrobials? Because there's a bit of confusion about this, and I just wanted to have an idea of what people think is, and it's one of the misconceptions that tends to exist out there, because it's not always clear whether it's us who are taking antimicrobials or taking antibiotics to fight an infection, or is it the bacteria or the viruses that we're trying to fight that are resistant to those drugs, or both. So that's kind of what I'm like to find out a little bit what you think is involved in this particular case. And again, I'm going to give it a few seconds for people to join in this interactive experience. This is you can't get more interactive than this, I guess, because I'm giving a virtual presentation, and then we're all using a different apps to try and connect. without me seeing who's there, which is kind of strange. So we have a few answers, and most people actually think, they gave me the right answer, that it's the bacteria, it's the bugs that are resistant to antimicrobials. Some of you think it's both, and I'm now I'm going to try and kind of explain why that's not correct, and it's actually just the bacteria or the viruses that are resistant. So it's when we're talking about antibiotic or antimicrobial resistance, the difference between those two terms is when we say antibiotic, we're specifically talking about drugs that kill or attack bacteria. When we're talking about antimicrobials, we're including in there drugs that will kill viruses. You might have heard about those recently, but we're also talking about drugs that kill fungus, for example, which are also a source for infections from bugs. And that what I'm going to say about antibiotic resistant applies to antimicrobial resistant as well. So in this talk, I'm kind of going to use the terms more or less interchangeably, but I just wanted to explain what those two things mean. And the reason why the bugs develop this resistance is basically what happens is that because we are overprescribing antibiotics or people don't finish their treatments, it's one of the topics that's not discussed enough, I think, is the fact that we overuse antibiotics in our agricultural setting, so in livestock and fish farming, and that's obviously goes into the food chain. So that means that we're going to get bugs that generate resistance there. So even if we tackle the overuse of antibiotics in humans, if we don't at the same time have a look at the overuse of antibiotics in animals, then we would still have a problem. Other potential sources have to do with infection control in hospitals or a lack of hygiene and poor sanitation. And one of the key things that I would like to stress, and people might be more might understand this a little bit more with the recent situation, which is we need investment, we need research into new drugs, and there hasn't been, specifically on the antibiotics. There's been very limited new classes of antibiotics that have been developed in the last 20, 30 years. So most of our antibiotics have been around both human and animal populations for quite a while. So the bacteria have been exposed to those drugs for quite a long time, which means they basically start developing ways to not be affected by those drugs. So we really need to develop new drugs and new ways of treating these infections. And as I mentioned, one of the ways that we get superbugs, so bacteria, I'm going to focus on bacteria from now on, bacteria that get resistant to antibiotics is one of the sources is from food, but also from person to person and contact. And as I said, I'm really glad because this has become a topic of that's more on people's minds and within the healthcare systems. There's been a change of how antibiotics are prescribed. People are becoming increasingly aware that they shouldn't just overuse antibiotics, and there's a good way of taking an antibiotic, of treating a bacterial infection. And just to give you an idea, that people do kind of search about antibiotics and want to be informed about this. This is just, the 100 there is in a year's period, we're looking from last June to this week, how often people search for antimicrobial resistance. So the 100 is the top of the scale. And it's kind of consistently something that people are interested in. And then, as Nicola said, something happened. This year, which means people are actually way more concerned about something completely different, coronavirus. And it was, that that conversation back in January now seems quite distant. And there was another speaker at the time, Angela, who's one of the staff at the Freeman Hospital here in Newcastle that is responsible for testing a lot of the samples. And she was asked a question about coronavirus and whether she'd had any samples. And I think at that point she had one sample from Scotland that she had tested, and it was negative. I'm sure the story is very, very different now. So there's been a shift in the attention of people when it comes to important health problems and what could be a global health problem. And yes, at the moment, the focus is on coronavirus, but there's been a lot of talk about the fact that our crisis of antimicrobial resistance is still there. It hasn't gone. And actually, the coronavirus crisis presents some challenges, some new challenges associated with it. So I'm going to tell you a little bit about COVID-19 and antimicrobial resistance, which we shorten to AMR. And WHO had a report, and they're actually quite concerned that the overuse of antibiotics. So what happened, particularly very early on in the pandemic, is that people came in with COVID-19. They were very sick, and as a sort of almost blanket decision, those that were more seriously ill were given multiple courses of antibiotics without basically the idea is to prevent secondary infections. There was, that's the usual course, I think, normally for people who present to hospital with pneumonia-like symptoms, because even if it's viral, it can create the conditions for certain bacteria to kind of infect the lungs and exacerbate the problem. So there was this overuse, or I wouldn't say overuse, but overprescription of antibiotics very early on. And obviously this has the concern here is that we will be selecting for bacteria that are resistant to these drugs. And when the patients that recover go back to the community, they could spread those superbugs and those that are resistant. And there's been some interesting discussions and opinions from both scientists and medical staff on what are the dangers and the opportunities. So there's a few things that could actually prevent the spread of multi-drug resistant organisms or multi-drug resistant infections. The fact that we're keeping our distance, the fact that we're washing our hands much more. The fact that medical staff and staff in hospitals and care homes are all wearing protective equipment means that we're kind of reducing the opportunity for these bugs to spread. But then there's other aspects because that could actually make it worse. And one of them is what I just said, the overuse of antibiotics and the fact that people who then recovered, if they had a secondary infection, if they had something else on top of COVID-19 that was resistant to antibiotics, then that can actually spread to other people, at least in their close. And as generally across the world, lockdown measures are easing up, we will have more contact. The other aspects that hasn't been talked about much is if you isolate huge sections of hospitals and intensive care units for COVID-19 patients, you have less ability to isolate other patients, and that will be particularly important for I'm going to tell you about for the work that we do. So there's a little balance here, and luckily, the people are thinking about this. The World Health Organization, WHO, has updated their guidelines when it comes to the use of antibiotics and the prescription of antibiotics to try and minimize the risks of COVID-19 actually increasing and making this an even more serious problem than it already is. As I said, the problem hasn't gone away. We just need to make sure that what we're doing to treat COVID-19 doesn't make it worse or doesn't bring some of the concerns that anyone who's working on drug-resistant bacteria has. will actually start happening earlier, because you're just getting kind of an explosive mixture of things happening at the same time, and that's the last thing we want, is something else on top of this, to exacerbate the problems, the global health problems. So that's where we are in terms of COVID-19 and AMR, and I'd like to tell you a little bit about my own research and the research we do here in Newcastle, in my group, and with collaborators both in the UK and across the world. And as I said, it's the bugs that are resistant. So there's lots of superbugs. Don't be fooled by the cuteness of these little ones. So there's actually a company that does plush toys, cute toys, based on different bacteria and viruses and cells. So these are some of their representation of drugs of bugs that are resistant to drugs. And this is my favorite one, which is called Clostridium difficile, or C-Diff. It's not purple and it's not cuddly. It actually can cause severe disease. So what happens is that if you're on antibiotics, that disturbs the good the good bugs in your gut. I'm sure most of you have heard about the good bacteria in the gut. So at least some classes of antibiotics that we use, or if not to say all, will target and they will kill good bacteria as much as they kill bad bacteria, because the way they work is that they'll attack specific parts of the bacterial cell, which are common to the good and the bad bacteria. So if you're on antibiotics, you're killing your good bacteria, you're creating the conditions for C. diff, the purple one here, to multiply, and they produce certain proteins which are toxins, and those toxins basically they make holes in your gut, and that means the milder symptoms are just diarrhea, which will generally, in a healthy adult individual, will just clear with time. But the problem is that it can actually lead to much more serious damage to the gut, and it can lead to death in some cases. And on top of that, if you get one infection from C. diff, you're 25% more likely to get a second one, for it to come back, basically. If you get 2 episodes, you're almost 60% more likely to get a third one. And if you get 3, you're looking at 75, 80% more chances of having a fourth one. So you have recurrent episodes, and there's reports of people who basically have diarrhea over the course of a year. And you can imagine what, anyone who's ever had diarrhea for a couple of days, imagine that constantly. You're depleting your nutrients because they're not being absorbed. You're dehydrated. There's a whole range of problems that even just having recurrent episodes of a mild diarrhea can actually be quite problematic. And one of the problems of C. diff is that it actually is resistant to most of the drugs that we have. So there's only currently 3 antibiotics that we can use to treat it. And that obviously, you were on antibiotics and somehow you got in touch with C. diff, and in order to treat the C. diff, you're going to take another antibiotic, which is going to kill, you're going to make the good bacteria even less likely to come back. And on top of that, there's only 3 drugs that we can use. And we're starting to see resistance to at least 1, possibly 2 of them in certain situations. So our options are narrowing considerably when it comes to how do we treat this bug. So, as I say, it's one of the, it's actually one of the 10 superbugs that has been identified of serious concerns in the next few years, both by the World Health Organization and things like the CDC, which is the disease control agency in the US. So, time for another question. So if you want to go back, pick up your phones again, and I'm just going to throw it out there. What superpowers do you think C. diff might have? How do you think it might actually survive so well, or fight off the antibiotics that we use? And yeah, it's open to your imagination. Just give me what do you think. How do you think it might protect itself from our attacks? But basically, that's how you have to think about it. It's kind of like a tug of war that we're playing with it. We develop drugs and they'll fight back and they'll find ways of defending against them. So there's no wrong answers here. And you can be as imaginative and as silly, or try and think of what if you were a bacteria trying to live in our gut, what would you do to be like super powerful and rule the gut, as it were? And I'm going to give you a couple more seconds to come up with some ideas of what do you think could be good strategies to do that. And hopefully there's a few ideas. So some people are saying it could disguise itself as the host cells. It could divide really quickly and could recover fast. Swapping of DNA is change the outer coat, changing the coat. There's very good suggestions there. Have an invisibility cloak. That's a good one. A protective shell, quick mutations. That's all very cool answers. So thank you for that. So one of the superpowers that it has, and before we go on and reveal what superpowers it has, just want to tell you a little bit more about the actual disease and how much it affects people. So there's this is data from the disease control agency in the US. So this is data for the US. So there's about 250,000 people who are infected with C. diff every year. About 14,000 deaths. To give you an idea, in England and Wales, which is the data that we have for the last data we have is actually dates a few years back, but we had about 3,000 deaths and 30,000 infections. And I know compare that to COVID, it doesn't sound very hard, but this is not sort of a global pandemic kind of thing. It's spread out. It's still quite a lot of problem. But I always like to go a little bit beyond just the statistics, because they can be just that, just numbers, and it's kind of hard to understand what's behind it. So just a couple of personal testimonies, and this is just someone who had C. diff, and it had those kind of recurring episodes, so it just kept on having those infections. And she was dealing with this for almost 3 years. And it's you can imagine that kind of how debilitating that can be. And this is another example of someone who is a healthy person, and in 3 months just their health really declined. So it can be not only in the sort of more severe acute cases, even in the less the ones that aren't fatal within a few weeks, or the ones that don't develop to such serious symptoms. It can be absolutely debilitating, and that's why we're interested in it. So we've had what are C. diff superpowers, and one of its superpowers, it can actually lay dormant. It can be sort of a bit like Sleeping Beauty, laying around, basically not doing much, and in this form, which is called the spore, it can resist a lot of the ways we would normal clean things. It can resist radiation. It can resist heat. It can resist things like ethanol, for example. So in this form, it can stay in the environment. And if you touch a surface that has diff spores and then touch your mouth, that's one of the ways that you would get C-Diff inside you. And if you were on antibiotics, or if for some reason your gut wasn't as healthy as normal, then it could flourish and cause an infection. And we're particularly interested. These are the spores. This is what they look like. So you have the inside of the cell, and then it has several protective layers that just keeps it kind of keeps it protected from all these different ways, the different sort of environment that can attack it. I should say, I haven't said so earlier, but C-Diff can't live when there's oxygen, so it's a very ancient bacteria, and it's thought to be 4 million years old, and there was very little oxygen at that point, so it evolved to have this dormant form as a way to protect itself, and it's because it can't. That's one of the reasons why it likes our gut, because our gut doesn't have a lot of oxygen, so it's a good environment, and it has a lot of nutrients, so it's a good environment for it to come back to life and cause problems. These dormant forms are one way to make sure that it can stay alive outside in the outside world, and that's one of its superpowers, is to be able to do that. So in my group, we've been and this is just to tell you what happens when those spores are made. And basically, this is normal shape of C-Diff, and it would normally divide sort of halfway in the middle. And when it's creating those dormant cells, those spores, it divides just asymmetrically. So you have a smaller cell and a bigger cell, and then over time, the bigger cell is going to kind of engulf the small cell, which is that small cell will eventually develop to become the spore, and this sort of engulfment, the way that the mother cell just kind of wraps around and protects the spore and keeps it inside, is one of the processes that in my group we've been interested in, and these are some. Abby, Adam, and Mochin have been focusing on that, and what we're trying to see is understand what's the force that allows it to do that sort of wrapping around, and what's the pulling machine? What is the kind of the mechanism for that to happen? And there's a lot of there's basically the machine is made up of proteins, and there's several proteins that we've been interested in, and we've come up with a way of how they interact by using microscopy techniques. So we're looking at the bacterial cells, and we basically delete some of these genes and then find out what happens to understand what they might be doing in there. We also we're a biochemistry lab as well, so we try and understand how they do their function and how they do their job, and we try and understand how they come together and how they might interact. And with combining all of those techniques, we have come closer to our understanding how the pulling machine comes together and what the different cogs are, and how they interact and how they work together to allow this kind of wrapping up. So that's one of the superpowers, and it's quiz time again. So if you want to go back to your phones and check the next question. Yeah, we're going to need a few. Yeah, everyone's few people coming in. I'm going to start the quiz. And there's no points, there's no prize. There might have been a drink if we were in a bar, but we're not. So I'm going to give you, I think there's a 15 seconds to give your answer. And time's up. So I agree. This is a fantastic trilogy of books. As you can see, that's what I marked as the right answer. And yeah, the movies are good, but the books are much better. So I would definitely recommend you to have a look. But you might be sitting there thinking, what is she on about now? She was talking about bacteria and C-Diff and superbugs, and all of a sudden she's asking about Lord of the Rings. What's that have to do with anything? And if you answer the next question, the answer will start to be revealed. So you have one more question to answer on the quiz. And this, if you have either read the books or saw the movies or both, you'd remember that one of the characters is Frodo. At one point, he is stabbed, but something protects him from that sword. And I'm just wondering if you remember what it was. So yeah, okay, most of you do remember there was a mithril coat. And those of you that haven't seen it, don't know what I'm talking about, you might be thinking, what is that? And again, what does that have to do with our C-Diff? And this is what the mithril coat is basically a sort of metal woven coat. So it's very hard, but it's very flexible and light as well. But it has the power to protect from attacks. And that's what C-Diff has. It has its own sort of, not necessarily an armor, because an armor is you kind of think of an armor as a bit more rigid structure. This is very flexible and it adapts to the same shape as whoever's wearing it. So that's what C-Diff has, and that prevents it. That allows it to be protected from attacks. And we're no longer talking about the dormant cells. We're talking about the cells once they're in our gut and they're causing infection. And we've been trying to understand this armor or this coat, this mail coat that Diff has, this protective layer, using several techniques. Macromolecular crystallography, as Nicola says, that's my expertise. And when we use synchrotron, so we use particle accelerators, so to use the powerful X-rays that are generated there. We can also use other techniques like electron microscopy, and the idea is that we're trying to understand the shape of the proteins that make up this layer, and we're also trying to understand what that layer does, and because it's not very clear. And Anna and Paula, who have been helping on this project, we finally determined the structure of this protein. As basically we know what it looks like, and not only do we know how the protein looks like, but we know how it comes together to create this layer that covers the whole cell and protects it from the outside, and creates that sort of flexible but hard, impenetrable protective coat that it has. And you can see that it has sort of these little tiles, and they all come together, and then you have something on the top that kind of creates an extra surface. And we're working with colleagues, namely Oichik and Jason from the University of Sheffield, to look at what this looks like in live cells. So what they did is this is basically the outside layer of a C-Diff cell, and we can use electron microscopy in particular ways to determine how it looks like, how the layer, which is called the S layer or the surface layer, looks like in the actual cells. And this is this kind of rough the map that we saw, and you can see that the structure that we have determined kind of fits there. Those triangles make one plane, and then you have these little ridges on top, which kind of would allow for interactions to happen with our gut. We've taken, together with collaborators both in Sheffield and in Glasgow, we've taken this one step further and trying to understand the actual function of this layer. So Philippa, who was in Glasgow for a while, she used mice, and mice don't get very sick with C. diff. So they get infected, but basically what happens is that you infect them with C. diff. You just give them the spores in their food or their water. So there's kind of not really any aggressive treatment done to them. And they'll lose a little bit of weight in the first couple of days, and then it just bounce back. And that's what happened with our mice. So when we infect them with a normal strain, they bounce back. And when we infected with a strain that was missing one of the bits, so one of those kind of little sort of ridges at the top, the mice weren't as sick. And this was despite the fact that, and just kind of reminding you of what happens when you get diff, this was despite the fact that the actual number of cells, of bacteria in your gut wasn't changed significantly. And the amount of toxins, which is what normally is thought to cause the problems by damaging the gut and the lining of the gut, that stayed the same as well. So this gave the first indication that actually the S layer, so the surface layer, is important for disease, and we're trying now to understand why that is, and that's going to be the follow-up work. And the idea is that because this layer is important for disease, but also is important for the cell, if we can disrupt this layer, we should be able to attack C-Diff in a much more effective way. So we should be able to develop drugs that, instead of killing the good bacteria, actually just kill C-Diff if you have an infection. And that would mean we have a targeted drug. There's less chances of it being able to develop resistance to those drugs, because we would be tackling something that's essential for the cell. And there's less danger for other bacteria that would also be in the gut to also be affected and develop resistance or share resistance, because it's something that would specifically just be affecting our C. diff cells. And going back to COVID and the reason why we're doing this this way rather than in the pub and having a chat, to COVID-19 and what specific dangers are there for C. diff and COVID-19. I mean, one of the problems is to do with detecting both of them, actually, because 10 to 20% of COVID-19 patients have diarrhea as a symptom, depending on the different studies. This is, it's all very new, so you know that a lot of the information is coming as things are happening, so it's hard to have a definite number. But there's clearly a reasonable percentage of people that have diarrhea as one of the symptoms of the COVID-19 infections. And in some cases, that's the only symptom. And so they wouldn't be diagnosed with COVID-19 because it's not kind of in the necessarily within the first thing that comes to mind when you present with just diarrhea. In terms of co-infection, there's some problems and some issues that are raised. One of them is, CDI is hard to treat, so if someone is already debilitated because of COVID-19 and then gets a C. diff infection, you might complicate increase the risk of death because you're debilitating that person's condition even more. But as I said, because they share symptoms, it might be really hard to diagnose either one. So if you're a confirmed COVID-19 patient and you get diarrhea, you might not be tested for C. diff, which might lead to problems, but vice versa, if you come in with diarrhea and you're positive for C. diff, people, it might take longer to realize that you also have COVID-19 and you need to be treated for that. Although at the moment we don't have treatment, but that needs to be taken into account in how the patient might be handled. So far, there's only been one study that looked at co-infection in COVID-19 patients. This was a hospital in Detroit in the US, and they found from the COVID-19 patients that had been admitted for a period of 3 weeks, 9 of them were co-infected with CDI. All of them had been on broad-spectrum antibiotics, either prior or on admission when they went to the hospital, and actually 4 of them died. So there's, that it raises the possibility that actually the outcome was worsened by the fact that they had C. diff as well as COVID-19. And this is true not just in hospitals. This seems to be happening in care home settings as well, which is obviously, as we all know, one of the concerns. And it's kind of more, I wouldn't say more serious, but it's more worrying for care homes and C. diff, because there's an overlap of the more susceptible groups. So traditionally, although this has changed recently, but traditionally the elder elderly are more at risk of a C. diff infection and definitely more at risk of recurrent infections. And that's the same with COVID-19. So you have overlapping susceptible groups, and you might have someone coming in who doesn't have COVID-19 and does have CDI, but you can't have patient isolation because, as I was saying earlier, the isolation capability of that particular hospital has been kind of taken over by COVID-19 patients. And as I said, the antibiotic usage is an increased risk for developing CDI and having those recurrent infections and complications coming from COVID-19. So we need to keep an eye on it. We need to see what's happening. We need to have an idea what's happening in the hospitals, what's happening in the care homes between the two of them, and as the WHO has recognized, we need to maybe there needs to be a shift and more care when it comes to antibiotic prescription for COVID-19 patients. And there's been a few recent studies in the last 2-3 weeks suggesting that there's actually a relatively low level of co-infections generally on COVID-19 patients. So the blanket use of antibiotics without a clear indication of a secondary infection should be taken into account in hospitals when treating patients. And I'll just like to finish. I mean, this is a long list of people who have worked with me here at Newcastle University and collaborators in Sheffield and Glasgow University, particularly Rob Fagan's group and Jill's group. And this was all started by when I worked with Neil Fairweather at Imperial College. And thank you very much. I have one last request to go to menti.com, and you can take the time of the break to answer that final question, which is just, I would, this would normally be a conversation. We'd be in a room talking, and I would just like to know if what you've learned today. So if you can leave a couple of comments there, that would be fantastic. Thank you very much. And I'm open to any questions.
That's great. Thank you, Paula. So before we go to a question and answer session, we will take a 5-minute break. So for everyone watching in the audience, I'm sure you have lots of questions for Paula. Please do put them into the chat box and I will ask them in a few minutes. So just to remind you again that there will be a little pub quiz during the question and answer session. If you would like to take part, you don't need a pen or paper. It's just a question and the answer, So it's fairly simple. But if you do have any questions about the talk you just heard, about the topic that you think Paula might be able to answer, then please do write your questions in the chat box, and we'll be back in 5 minutes. Hi everyone, welcome back. So now we're going to move into the question and answer session with Paula. So firstly, thank you very much for your talk. It was really interesting and certainly good. As I saw the talk before the coronavirus epidemic, seeing the connection now is really good. I'm sort of getting the full picture, which is interesting. So we've had a few questions come through on the chat. A lot of people interested with how this links to the COVID pandemic. So one person was asking, and I think you briefly touched on this, whether you think the C. diff infections will have decreased because of the COVID-19 pandemic as hygiene has increased, more washing hands and things like that.
So as I said, there's only one published study of C. diff and COVID and any correlation or any sort of, it's not even the number of cases have increased or decreased. It's just looking at co-infections. That is one of the questions I think we need to start answering and start having a look. I think yes, obviously washing hands and people being in isolation should help, but then there's other aspects like the fact that you might have patients who could be colonized with C. diff, not in isolation, next to patients who are being treated for something else who are susceptible to C. diff and the mix. The short answer is we don't know yet. There's anecdotal, I've heard from colleagues who work in hospitals that it might actually have increased outside COVID-19 wards because of things like people were thought to have COVID-19, they were sharing, they actually had C. diff and they passed it around. And because you're talking about more elderly patients that are more likely to have C. diff and have problems, it actually makes the situation worse. So it's going to be a balance, but we need to keep an eye on it.
Do you think over the long term, obviously, with the situation as it is, people, I think, in general have become more aware of how easily you can catch things and spread things and how important washing your hands and doing it properly, I think for a lot of people have realised, actually, I probably don't spend 20 seconds washing my hands and things like that. Do you think in the long term we'll see decreases over time?
I think that might be one of the positive effects of this, yes, in the long term, that you actually do the increase in hygiene, so outside hospital settings, in communities, we're washing our hands better, that surfaces are cleaned, that might actually reduce the exposure of people to it. So in the long term, it could actually be. But what we saw, and it's an interesting question because the epidemics as to how C. diff spreads, we had a big outbreak in 2007 in worldwide, and it was a huge spike, and then new cleaning measures were introduced, and that it meant that the cases decreased considerably. But in the last few years, we kind of plateaued at a level. So the question is, how much is hygiene on its own going to get rid of the problem? And again, it goes back to the farming issue. For example, there's been epidemiological studies done in some countries, namely there's one in the Netherlands where they found other animals get infected with it, and pigs is a common one, and they found that the strains that were infecting the pigs were the same that were infecting the pig farmers. So it's again, washing your hands would be good, but you need to think about it in a much broader view. So I think it should help that we'll be more aware of it, and just our habits are going to change slightly. But whether it's going to completely get rid of the problem, I don't know.
Yeah, hopefully habits will change.
Hopefully this new way of living. Hopefully that will stay.
Is there any way sort of retroactively to test if you've had C. diff? Does it stay in your body? Is that why you're more likely to get future infections, or is it? Can you tell you've had it and therefore be put at risk of getting it again?
We don't know. So there's another thing. There's another superpower that diff has that I didn't go about. That going to talking about it, which is as many other bacteria, it can form biofilms. So this is kind of basically a group of bacteria and different materials just coming together and creating sort of a mass, rather than individual cells swimming around, and that creates a sort of a protection and a hiding place. It's like it's kind of like they make their only sort of little cave where they're hidden from the immune system or from antibiotics, and that's one of the possible ways that you might get recurrent episodes. But we're not 100% sure whether when you get recurrent episodes, is it the same strain that you had initially, or is it a different one? So you get infected again, and there's reports of both. So I think it depends on it. Might be the case that it depends on the person and on the strain, on the particular conditions. Why are you so much more susceptible is again a question that we don't know the answer to at the moment. But a lot of us do ingest diff spores. They go through our gut. They can't germinate. They just come out the other end, and we have no problems. So we're constantly in contact with it. Is just the fact that you're on antibiotics and it kills the good bacteria makes you susceptible. There's a few other things. So if you're immunocompromised, would also, just generally with any infection, would make you more susceptible. But that's the key thing, is if your gut, good bacteria in your gut, are not there or have been affected by some way, then you have an increased possibility. So it could be linked to that. We still don't know the mechanism.
Right. Another question we had, I think very much relates to the current situation, to C. diff and other bacteria as well. Is it possible to gain antibiotic resistance from the increased use of hand sanitizer?
So the hand sanitizer, the way it kills them is alcohol, and alcohol can affect the way that their outside surfaces behave. And I mean, there's a whole range of ways that alcohol kills bacteria. I'm not going to go into the details, but that's the key thing. One of the ways that diff protects itself from the alcohol is by creating those dormant cells, which are resistant to it. So there are some bacteria diff is not the only one, who are able to do that, who would not be killed completely by the alcohol. Developing mechanisms against the hand sanitizer. I don't know. I'll have to look it up. It's a good question. Are there mechanisms to?
I know certainly. I've seen a lot of people really opt in for the antibacterial hand sanitizers in the hope to avoid COVID, and trying to understand or translate that viruses and bacteria similar in some ways, but also very different in others.
Yeah.
And how going for that antibacterial soap, things like that, just general increase in use of things like that, do you think that would increase resistance as well?
Yeah, it can, because basically what survives if you're using those things is something that, I mean, some of these antibacterial, what they use is either ethanol or bleach or kind of derivatives thereof, but some do use specific things. I mean, they don't use antibiotics per se, but yeah, it could have, you could be making things more complicated and worse, yeah. But yeah, I forgot to say that. Yeah, thanks for a reminder. Bacteria and virus are very different things, and one doesn't kill, what kills a virus doesn't kill a bacteria, apart from a few things like alcohol. But yeah. When it comes to drugs, they're very different kind of drugs.
We can all appreciate. Another question we had, talking about thinking about how bacteria sort of mutate and gain resistance. So after thousands of generations, can bacteria lose resistance to antibiotics? And as a result of this, would older antibiotics become useful again in the future?
I mean, so that's why we generally talk. That's a very good question. And yes, if the evolutionary pressure that caused a certain mutation which allows them to be resistant to a specific type of drug is no longer there. So if we're not, if the bacteria aren't being exposed to that drug, the pressure to keep that isn't there. So it might be lost. But one of the issues there is that we talk about classes of drugs, in the sense that certain antibiotics will kill the bacteria in a specific way by binding to certain proteins. And even though the drug itself might be different, and so different mutations might mean different resistance. I mean, the more resistant and the multi-drug resistant organisms, they tend to have mutations that allow them to be resistant to a whole class of drugs. But that is one of the things that has been tried. So looking back at older antibiotics that we haven't used to tackle multi-drug resistant bugs that we have now, with limited success as far as I understand. But I think there's been a couple of cases where, a specific version of a drug that had stopped being effective against a certain bacteria was effective against another one. So it can theoretically happen, yeah. It's a good suggestion.
But probably best not to rely on it, I guess, in the long run.
No, we definitely need to kind of pursue new ways.
Yeah. Another question we had, someone asked, are there any links between C. diff and TB?
Oh, I don't know what they mean in terms of co-infections, or it's kind of hard to answer, but not. I mean, not.
Yeah.
I think if you have TB, you would be taking. So one of the links is that if you have TB, the antibiotic treatment for TB to kill Mycobacterium tuberculosis, which causes tuberculosis, is I think 3 or 4. It depends on the cases. It depends on the strain, but up to 4 different antibiotics for a long time. So these are kind of between 3 and 6 months antibiotic course treatments. So obviously during that period, you would be susceptible to C. diff. I don't know off the top of my head if people have looked at the incidence of C. diff infections in TB patients, but yeah, that could be one of. I mean, if you are on antibiotics for that length of time, you would be more susceptible. It's one of the many reasons why people need to be kind of shielded and protected when they're doing those treatments. I don't know if that answers the question, sense. But if they want to follow up,
I'm not seeing any update, but. One question I had, so as we've seen with this pandemic, and obviously it happened very fast, it needed to be treated very fast to stop things getting bad very quickly. And certainly I've noticed across the research community, people coming together so rapidly in terms of collaborating, exploring different areas, publishing papers. The speed at which papers are coming out related to the coronavirus is just mind-blowing. Do you think with all this collaboration that's happening amongst the research, like sort of virus research community, do you think you could see something similar in the future working towards tackling antibiotic resistance?
Well, yeah, I mean, it's been fantastic to see how open the scientific community has been about the whole coronavirus and the studies. There's the caveat that sometimes some studies have come out that didn't go through the normal procedures or just jumped through a few hoops in the kind of hope to publish. But they've been corrected, and they were published in record times, but they're being corrected in record times as well. So that kind of shows that the system is working. I have to say that the C. diff community is actually specifically is very open and actually very collaborative. It's one of the great things about studying this bug, which it's a medical problem, but it's also scientifically quite interesting because it does a lot of things different from other bacteria. So that sparks our curiosity. It's a very open and collaborative community. I think we have lessons to learn and how we can be even more open and I think this crisis will have increase the speed that we do open science, for example, and share data even before it's published a whole lot more easier. And I think globally, in terms of the antimicrobial resistant people who work in the field, again, it's generally quite open and about sharing, but not at the scale of what we've seen with coronavirus. And I hope will be as good going forward as the virologists, epidemiologists. I mean, it's been a huge range of people who've worked together and found new collaborations and new ways of talking to each other. And I think we should learn from that.
Yeah, definitely. And as you said, it's really, a lot of the stuff is really positive, how quickly people are working together, but there are some drawbacks, information being broadcast out there that hasn't been confirmed or peer reviewed and then been taken up by the media and things. But I suppose the more it happens, hopefully, the better we'll get at it. And other areas I think antibiotic resistance will benefit.
Yeah, I agree, and I think people will hopefully generally the general public and the non-scientists will understand the process a little bit better, because sometimes, I think it has been clear that sometimes the media picks things up without giving that caveat that any scientist would say is just kind of like, this is what we know now, it might change in a week, a month, or a year, or 10 years. And I think this has kind of brought that to light a little bit more. So hopefully it will mean that there's a bigger understanding of the scientific process as well as the science itself going forward.
Yeah, definitely. And as a scientist myself, I include that caveat anytime I speak about my work. This might not be true. It's a nice little safety net you can throw in just in case. In terms of what you've seen globally, certainly in resistance and how bacteria are changing, is there any difference between sort of countries that have different healthcare systems? So, for example, in the US, their healthcare system isn't socialized. A lot of their doctors do prescribing based, not based on financial incentives, but that is certainly something that they deal with, and it has been not known, but said that perhaps some doctors in the US might overprescribe because of that. Do you think, are there any differences in antibiotic resistance between, for example, the UK and the US if they're prescribing more and having the after effects of that?
I mean, the World Health Organization is monitoring this, and it's kind of a voluntary country reporting system. So in terms of antibiotic prescription usage and the finding antibiotic resistant strains, and there are obvious hotspots where antibiotics are more widely available, then you're going to see more resistant strains coming out and more difficult to treat infections. A normal example is countries like India, where everybody knows that it's relatively easy to just buy antibiotics over the counter, but even in differences in countries like US and Europe in general, there are differences, and how that's why there's a huge campaign on stewardship, that is how antibiotics are used, and there's a huge pressure for doctors to change the way they prescribe antibiotics, but also for people to change the way they take them, and not go in and say, I have someone who goes in with flu-like symptoms and asks for antibiotic from their doctor, because as we were saying, flu is caused by a virus, nothing to do with a bacteria. The antibiotics are not going to do anything to it. So I think it is improving, and there was the latest report from the WHO showed an overall reduction of the problem in terms of over prescription, and so I think it is changing. There's more awareness. People are. I certainly in conversations with friends and family who don't know that much about it, I know they're more aware of it, if nothing else because I've kept hammering the idea for the last few years. Just don't take antibiotics if you don't have to, joke aside, I think there's more awareness, and definitely as a patient myself going to GPs, I've seen a change in their approach to it. But yeah, different health systems and the incentives to prescribe drugs when they're not necessarily needed is another issue that's part of a multifaceted campaign to tackle this. So it's not just about the science in finding new drugs; it's also how we keep the drugs that we do have useful.
Yeah, definitely. And I think it's good to know. I think prescribing by sort of GPs is changing. I think a big change has happened in the veterinary community as well. I think prescribing antibiotics has been vastly reduced to sort of farm animals and things like that. I think just knowing what to do. Yeah, if you're suffering from a virus like a cold or a flu, don't seek antibiotics because they won't work. If you are prescribed them, finish the course. And if you find any left over, well, don't flush them down the toilet. I think that was one of the things I learned, that there are special ways to get rid of antibiotics. Just putting them in the bin or flushing them down the toilet is bad.
Because it basically goes in, for example, diff lives in the soil, so it goes, and many other bacteria do. So, or the spores reside in the soil as well. So it's the issue with the farming is a big one, and with conversations currently between the UK and the US, and the possibility that meat from the US might come to this country much more freely. It's a known fact that the level of antibiotic usage in farming in the US is much, much higher than anywhere in Europe. So theoretically, that could bring the problem here or exacerbate the problem here, because you might get you might be bringing the meat and some antibiotic resistant bacteria along with it into a population that hasn't been exposed to them. So all those things need to come together, and the vets have as much of a big role to play as the doctors, and all of us.
Yeah, definitely. Yeah, everyone's involved.
Yeah.
So one last question we have, quite a good one to end on, but it's very broad. I'll let you interpret this as you see fit. So someone has asked, what is one thing that you would change? I can limit that in saying maybe in terms of your research, what you would hope to see or changes to be made, but what is one thing that you would change?
In terms of our research, I think what we're aiming for and what we would like to change is the way we, the way that C. diff can be treated, and having those targeted drugs is the holy grail kind of thing. So it's changing the way infections caused by C. diff are tackled is the one thing I would change. I would change it from, let's just use broad antibiotics to let's see if we can get drugs that kill just that. I mean, there's a whole range of other things that I could say. If there's one thing I would change, that's a good question and a tough one, but I'll do it focused on the research that we're trying to do.
That's a very good plan, and hopefully you'll be back in the lab soon enough and continue with that research and get lots of useful results that we can all benefit from.
Thank you.
I think that is all of our questions for now. So I'd just like to end on obviously saying thank you so much for sharing your work with us. For those in the audience, or indeed Paula herself, if you want to watch this talk back, then you can on our Palace of Science YouTube page. You can also, if you visit the YouTube channel, you can also see all of the talks we've done previously in the last couple of lockdown. And you can also tell your friends if they want to catch up on this talk to see this there as well. If anyone wants to get involved in Palace of Science, there are a number of things you can do. So firstly, you can visit our website or our social media pages. We're on Facebook, Instagram, Twitter. And please suggest any speakers that you would like to see or any topics that you would like to hear from. We, of course, like to keep our audience happy. And it also is a big help to me, as it saves me from choosing all the topics that only I want to know more about. So please visit our pages and suggest things that you would like to hear. Secondly, if you want to support us financially without having to donate anything, then you can sign up to this website, the Easy Fundraising site, and select Palace of Science for your donations. So essentially what this does, you can continue all of your shopping online as normal. When you make a purchase from any of the listed retailers, they send a small percentage of that purchase value to us. You don't pay any more, but the retailer donates some money to our cause, and we will really benefit, so that would be great. And finally, if you'd like to get involved more in Palace of Science itself, then you can. You can go to our page and sign up to our volunteers group. As I mentioned before, we are a group of volunteers. All our events are planned and brought to you in our spare time. So the more people we have on board, the bigger and better events we are able to run. I'd like to make it clear that you don't have to have a scientific background involved in Palace of Science. All of our events are specifically targeted to non-specialists. So having a non-specialist on the team can really benefit us. But obviously, if you are a scientist, then all of the knowledge from your field will help us as well. So we're happy to accept sort of anyone and everyone, show you can be of great value. Finally, if you don't want to get involved, you just want to see more science, then we're going to be bringing some more to you. So our next SciBar is on Wednesday, the 22nd of July at 7:30. So the usual time is 7:30. It's another excellent researcher in psychology from the University of Sunderland, and she's going to be talking to us about body modifications in the form of tattoos, which I'm really looking forward to. So do tune into that. And if you can't wait a whole other month for another event, then you're in luck. In 2 weeks' time, on the 8th of July, we have a brand new event. So Palace of Science presents. It's going to be a space for one-off events on a niche subject, or in this case, a little mini series that's going to take a deeper look into specific topics. So on the 8th of July, 2 weeks today, we will kick off our Diversity in STEM series. This series is going to take a closer look at the history and the practices, and most importantly, the people behind the fields of STEM as we know them today. So our speaker in 2 weeks is a PhD student in physics at Newcastle, although she's actually going to be talking to us about something separate from her PhD, although linked, and this will be the Forgotten Women in Science. Her talk actually came really highly recommended from a number of other people, and so we're really excited to hear from her. So that is everything from me. Again, I would like to thank Dr. Paula Salgado for an excellent talk. Hopefully you can join me again in 2 weeks' time for our Forgotten Women in Science talk, and if not, on the 22nd of July for the next SciBar. So in the meantime, enjoy the easing of restrictions, but obviously be careful and stay safe, and we will see you next time. See you later.